Announcement time. I messaged you with a couple more of these PowerPoints because they're just the ones we're using. They're just too bulky to include. And we're going to pick up where we left off there. I'll just put it up there. A couple of things. Exam wise, remember it's a week from today. Again, please, only pencils. I'll be remarkably upset if it's in pen because that produces a real scoring nightmare. So number two pencils, mechanical pencils, don't care. That type of thing. And again, the other thing I will tell you, I will not be here the following class day, which is the Friday. So the exam is on the 1st. I will not be here on the 3rd. I have to be away. So most likely I'll have to do a recorded thing because I'll be traveling, and it's almost impossible to get a signal in a vehicle trying to record something in real time. Nevertheless, so be aware of that. And again, what I will undoubtedly do before you return, I will most likely have the grades posted. Please don't ask me about points. Don't ask me about any of that nonsense. The only thing that matters is the letter grade that I post. I don't care what anybody tells you, have to have an 80% or whatever. I don't want to hear it. My responsibility is to give letter grades, period. You have to get a B in a class. Some people do have to get B minus works as a B. Effectively, the range for that is 80 to 82% if you want to get. So if somebody has an issue with that, a faculty issue, have them contact me. They won't. Okay? Please. So I don't want to hear that, I had this many points or that many points. Inevitably, that requires some degree of scaling or, quote, grade adjustment or, quote, curving. Tom will say exam 1 with scale, and then you'll see whatever it is, B minus, B plus, A minus, C plus, whatever. All of the above. If you want, we do not look at exams in class. We don't go over them step by step. That's what office hours are for. Make an appointment, you want to see it, fine. Figure out what you got wrong, fine, I'm good with that. We've got too much material to cover to waste more than the minute or two I'm having talking. That's all. Moving on. Good to go. I know you're upset not having me here on the 3rd. Please. Don't want you to go. I don't want to make this into a big issue. Surprised you're not up and applauding. So here we go. This is just to quickly review. I want to finish up all about vesicular transport. Effectively, it's a very, very brief review of what we did before. The whole idea of opening and closing membrane is so important. It requires energy. That's why it's part of active transport. We package things in vesicles to protect them. We'll start to probably introduce the old parts of the cell my way as we get toward the end of today's period. The whole idea why you have this elaborate thing we call the endomembrane system is to protect things. The inside of the cell is nasty. It's harsh. So whether we're bringing something in or we're spitting something out, the membrane is there to protect it from other things that are going on in the cell that might disrupt it, okay, or damage it, or expose something that's nasty to the cell, like a bacteria or a virus, to the interior of the cell and damage it significantly. We'll look at that a little bit today too. So as your endocytosis bringing in, where exo is transporting it out. Transcytosis, you see more in AP2 when you talk about digestive and urinary. Effectively, it's just transiting through the cell. It's protected in membrane, but not being interacted. So nearly anything that we bring in by endocytosis, it will effectively, other than transcytosis, which just transits through the cell, basically that liner is nothing more than a passageway until it gets in the bloodstream. That happens in digestive. Okay, that happens in urinary. Other than that, anything else will fuse with a lysosome effectively to determine whether it's something that should be digested and utilized, or if it's an invader to be destroyed and the remnants gotten rid of in some way, shape, or form. That's what I mean by digestion and defense. And I'll reinforce that when we get to the part about the lysosome. So even though they're phago and pinocytosis, one's solid, one's liquid. So pino, liquid or oils. Phagocytosis, implication is solid. They all involve receptors. Basically, for that pocket to be created, something has to come in contact with some type of receptor protein. When we look briefly at receptor-mediated endocytosis, it's a very specific construction of receptors, and it really has some real significance vis-a-vis people who trouble with have to take medications to control it. So all that's coming your way. Vesicular trafficking is just basically to understand that nothing is random inside the cell. There's a really elaborate network of roadways and passageways that, interestingly, bless you, can be constructed and deconstructed, okay, throughout the cell. So a roadway, if something brings in, a roadway will develop, it'll bring it to a destination, the roadway will disappear, and it'll form somewhere else. So the whole idea, if there's a hallmark of today's presentation, appreciate the dynamic nature inside the cell. Things are always changing. So endocytosis, yes, there's proteins that are there. They act as receptors. The substance has to some way, I don't know if bind suggests something that's a hard, bound to, like tied to. I don't know that that's really right. It has to be able to at least trigger that receptor or series of receptors to create that sort of pocket that eventually will surround it with membrane. It's very unique. The big problem, bad guys, bacteria, viruses, toxins, taking over this mechanism. And that's something you see more examples of in microbiology down the road. But understand unfortunately, this is how a lot of things work. In the biology, in our universe, viruses are not living things, but bacteria are. But the mechanisms are very, very similar. So they can utilize our mechanisms to their advantage. If you want to think of it that way, without thinking that the bacteria and the viruses have a plan. Okay? And so what will happen is either going to go right through or it's going to fuse with the lysosome and be interacted with in some way, shape, or form. Went over this with you before. Not going to go through all the slides. Yeah, there's coats there and it sheds them. Who cares? But basically, it's either just going to go right through and be expelled on the other side, typically into the bloodstream if it's indigestive, or it's going to be interacted with in some way, shape, or form. It'll either be digested and used as building blocks and be moved to other areas, either inside the cell or secreted or given off outside the cell. Or if there's waste materials from something being destroyed, it'll also be spit out eventually. So it's a well understood process. So without going over it again, so phago, eating, just the name, so it implies solids. The whole idea, these effectively almost all of these cells, and white blood cells in particular, have what's called amoeboid motion. When we get into the interior, that's what we call the cytoskeleton of the cell, you're going to see there are things called microfilaments and microtubules. Imagine. So amoeboid motion is the analogy would be like a baby scooching around on the floor. It slides around and slides around. The cell is filled with cytoplasm. The membrane is fluid. So if we can somehow push the membrane to one side, the cytoplasm will flow to fill it. And so imagine that this is the membrane. If you have an arm, the cell has an arm like this, that arm can disappear and rebuild on the opposite side. So these what they call microfilaments can disassemble and basically be reassembled over here and push on the membrane and the cytoplasm will flow. That's kind of in the same way that it goes to enclose something to bring it in. It's the same mechanism. These tiny filaments are developed and they're called microfilaments. You'll see them. I have a very good animation when we get to the conclusion of this unit, probably at some juncture before we do the cell cycle, to kind of show you how this works. My animation's been around for a long time. Okay, you can find it on YouTube fairly easily, I'm sure. So that's the idea of amoeboid motion. Flows into these temporary extensions and it allows the cell to creep or scooch around like a baby would be able to do it. Here's what basically happens. The receptors, such as they are, I think bind is not, as they demonstrate here, may not be a really great way to express it, but it induces this sort of sac to form around it. And membrane is conserved. If you're bringing in membrane this way, when we spit it out, it's not like it disappears. The membrane that was used to bring it in and basically remove from the outer membrane of the cell gets reincorporated, as you will see when we spit it out. So it's not like a cell that's eating is all of a sudden going to shrink. Okay, it's not. Okay, because it's a continuous process. That membrane that was used to make multiple of these things is going to be reincorporated on, so to speak, on the other side. That's there. Pinocytosis, fluid, cell drinking, sampling the environment. Okay, and when it fuses with a lysosome, we call it a phagolysosome, a fancy name that I won't ask you about it. That they call these endosomes, phagosomes, endosomes. If that language appears someplace, and I'm sure it's not on, and I'll try to take it out of my exam. If it is, I'll make sure we address it before. It's certainly not on the exam you're taking on a week from today. We do this all the time, sampling the environment. Our immune system is constantly sampling the environment. I mentioned it. In the skin, you have these things called Langerhans cells. They're what we call dendritic cells. They have long, multiple tentacles. And what they do, any place we're exposed to the outside. Your liver has something called Kupfer, capital Kupfer cells. Same deal. You have other ones all along your immune system. They're constantly doing it. It keeps your immune system in tune. It's checking the outside to see if, we have an invader, we better react with it. It's like any other system that you have. Think of any analogy. There's a thing of use it or lose it. And that's why that's such a really important system. And down the road, when you hear about micro, there's something that historically we have recognized now called the hygiene hypothesis. And effectively, what it was is the mistake that people make because we're excessive bathing, excessive hand washing. Okay, you're denuding or removing things that are important, that are there. We need to be exposed to things, immunologically. It's kind of some of the misunderstood things. I don't want to get into it. That's for micro about COVID. You all are part of that, right? At home, going to school with the masks on, all that nonsense. And I mean the word nonsense sincerely. That's my opinion. Don't get me started. So that's why you need to understand why these things are important. We go down the road. Here it is, bringing in liquids, how much fun. Receptor mediated, and this is something I'm not going to ask you about, but these are called clathrin. I save that for my advanced students. Clathrin coated pistons. It's a very specific chemical, a very specific protein. Okay, and it's designed for certain things. For instance, insulin. Unfortunately, viruses, diphtheria, cholera toxins taken in this way. So, as someone in your family, typically an older person, is taking medications called statins. Okay, they're designed for people who have high, they call it bad cholesterol. The LDLs, or low density lipoproteins, are really what they're referring to. When you have too many of these in your bloodstream, whenever there has been damage to and your blood vessels internally get damaged a lot. They get repaired very easily, but if they're damaged, sometimes they become inflamed, and then these fatty particles can become attached to them, worsen the inflammation, and eventually calcify. That's called hardening of the arteries. Something called atherosclerosis or arteriosclerosis. The process is understood. One of the things that we have are these receptor-mediated endocytosis. We're bringing in these low-density lipoproteins are fats that we can use. If you are genetically, and it's a hereditary thing, if you have a fault and these receptors don't work, you have too much in your system. And that's what we call it a hereditary lipidemia. Lipid for fats, emia means in the bloodstream. So if you have too much in the bloodstream. So with people like that, a lot of these quote unquote cholesterol medicines are designed. Typically the way to deal with these things, most people have problems with this because of lifestyle. They're very inactive, they have a terrible diet, they smoke, they drink. Pick a factor that's controllable. Okay, usually that's the underlying region. Those medicines are used for those people just to sort of limit the problem in the hopes that you can get a handle on it. I think it's overprescribed. That's my opinion, but nevertheless, there are people who really do require it. So, and there's a lot of things that fall into this period. For instance, diphtheria, if you did not know it, and you'll learn about this in micro as well, that every time you get a tetanus shot, you're getting immunized for diphtheria. You're getting 3 immunizations once. It's called a trivalent, means 3. Diphtheria, pertussis, whooping cough, and tetanus. And you're getting those mostly as preventative natures. If you really have been exposed and you're at risk of developing it immediately, there's other means that you have to employ that are there. Okay, we're not going to get in for this. We're not going to get into caveolae and the different. We talk about that a little bit in smooth muscle, but again, this is, for instance, tetanus toxin. This is the way it can enter. So there's a lot of the bad guys, so to speak, toxins, which are poisons, viruses and bacteria. All of those they exploit normal mechanisms. Might be the best way to express it. Here's what it looks like. Here, those lipoproteins, those bond directly and induce. It's very, very specific to form and bring this into the cell so it can be processed. I mean, that's really I think that's the fine distinction of specific receptor mediated involving those clathrin-coated pits. Okay. Exocytosis is even more interesting. It's how I mentioned before, when you bring something in, okay, it's processed, utilized, destroyed, whatever, and the remnants are spit out. And a lot of times in our cells, like a pancreas cell is making insulin. Insulin's a protein that has to be encased in a vesicle and spit out. We call them secretory vesicle. Secreting means releasing something. So whether you're perspiring, that came from a secretory vesicle. Okay, perspiration came out of that gland that you learned about undoubtedly in lab, or the oil. Here, that cell has given off some type of product and secreted it or released it in such a way that it'll either go throughout the body or go to the external environment. So it's called a secret. You'll hear chapter and verse about them, particularly when we're talking about neurotransmitters. This is the example I'm going to give. Mucus, an example of that in your digestive and respiratory tract. They're all exocytosed, spit out of the cell. It involves something called snare proteins, which you're not going to have to know about right now. This is kind of I'll give you through the passageway. This is the vesicle. It's got something in there to be spit out. That vesicle has a specific protein, and it's called V for vesicle snare, and T technically for a transporting snare. You don't have to know those. But what happens is, as they get close to each other, they basically, the snare is where they get their name, they intertwine. And in doing so, it pulls the vesicle closer to the membrane. As it gets close to the membrane, it induces a little opening or pore to form to the outside of the cell, and then subsequently, as the membrane incorporates with the existing membrane, effectively it spits the particles out. This is exactly what it looks like, magnified 100,000 times. So your cells are doing this constantly. You're thinking, your cells are releasing something called neurotransmitters. You're moving something, you're taking notes, your muscles are being triggered by those neurotransmitters. Interestingly enough, this is one of the areas where toxins interact. There's a toxin that stops those snare proteins from intertwining. We actually this is perhaps one of the strongest toxins made by bacteria that we've ever encountered. And yet now we refine it and take extraordinarily small amounts and utilize that to selectively paralyze muscles. Anyone wish to venture a guess? Fellow in the earlier class got it correct. You're not guessing. Why would you inject somebody with a toxin? No idea. If I gave you a hint, would you be willing to try? If it was used for cosmetic purposes. See, she got it. You were thinking it, I know. Botox, botulism toxin. Botulism is a deadly killer. If you don't know it, in the carpet now are what are called botulism endospores. There's tetanus endospores. They're not by themselves. Okay. These are eventually they have the capability under the right conditions to make one of the strongest toxins. These are neurotoxins, meaning they poison nerves. The way they poison the nerve has something called, and we're going to learn about this at the neuromuscular junction. Your nerve sends out a chemical called acetylcholine. It's a neurotransmitter to your muscle and says fire. If the acetylcholine, these little red balls here, can't be released, then the muscle can't fire. When a muscle can't move or change shape, we have a word for it: paralysis. It lasts for about 6 months, and then they're used not just originally for cosmetically, and then people use it for everything like hypersecretion. If they had like body odor, and they would inject their headaches, they would inject there to try to release muscles that might be causing like migraines. So there's a lot of usage today for Botox. But I still think the cosmetic aspect, it lasts for about 6 months, and once people start using it, they have to continue to use it because those wrinkles will come right back a little bit. You can see I didn't have that done for my blepharoplasty. My daughter managed one of those places where they would, and her task was to write every month $60,000 to $100,000 worth of business from wealthy individuals, mostly ladies, you know, keep looking, great. So it's really cosmetic. I mean, it's fine. You got the money, you want to spend it on your appearance, God bless, go ahead. I'm good with the wrinkles. That's right. I haven't had any work done. It's okay. My wife and I, we figure if we're getting old, we're getting old. Can't change that. Moving on. Don't tell her I said that. She comes in. Don't. He used the O word. Zip it. Nevertheless, this is what it looks like. We did the resting membrane potentially. You don't have to go crazy with it. Go watch the animation a couple of times. How it got to voltage, polarized, da. The big player right here, potassium. Because there's more potassium channels, and when there's irregularities in potassium, as I've said a couple of times, big problems. Effectively, why the inside of the cell is so negative, it has a ton of proteins concentrated. You have proteins inside the cell, and they're contained by the cell membrane and the various organelles. The ones in the cytoplasm, this is why I just sort of overwrote it, but effectively, they're organic acids. You remember, you don't have to know the chemistry. When you have, and something else is attached to it, that C double bond OH, what happens in the liquid of the cell, it's an acid. It gives up a proton. The hydrogen ion goes over here, H plus. What's left of that C double bond O is a minus sign. So all the proteins in the cell have a barely, and they're so large they can't get out, they create the interior of the cell has a minus charge. That potassium is attracted to come in, okay, and it has, because of all those leak channels, it has, it's even pulled back in, but the entire, even if the potassium escapes, it still has a substantial negative charge. It just does, even though there's more potassium on the inside. So the potassium is pulled back in by that negative interior, and again, the negative, even after it's escaped, that negative interior reattracts it back in. Okay. So I mean, and we established that gradient around minus 90. Remember that's from the illustration, from the animation rather. The sodium comes back in because there's less channels. Remember, there's always more potassium inside the cell, more sodium outside the cell. That's what those pumps do. All our cells have those sodium potassium pumps. The more active the cell, the more active the pumps are, the more energy it's spent. And what are they doing? They're basically keeping that gradient intact. They're pulling potassiums back in, they're kicking sodiums back out. It's the whole idea behind this. Chloride doesn't really influence, and you'll see it, okay? The interior, it's only along the membrane. I'll show you the illustration. It's right along the membrane. It's right here where you have more positive and more negative because of those leakage channels that are there. In the exterior, In the extracellular fluid, it's negative. I beg your pardon, it's neutral. It's neutral. The chloride neutralizes the sodiums and potassiums that are there. On the interior, down within the cell, the proteins, they neutralize all those potassiums. So other than right along the membrane, it's just in that location where we see the polarity. But because that is there along the membrane, again, in excitable cells like nerve and muscle, it can allow that membrane to quickly flip-flop the charge. It's almost, and again, that sets up the chain reaction that we're going to study in muscle and nerve physiology. I've got to set the framework for this, or it's going to be very difficult to process exactly how it works until you understand the basis of resting membrane potential. Down the road, we'll talk about chloride moves. There are certain medications, there are certain neurotransmitters that actually let chloride go in, and they actually make the interior of the cell more negative. You might be interested to know that's how something called alcohol works, and why it makes nerve and muscle cells less able to contract or fire is because it depresses the action, it lowers it. And you'll see how that works when we get to it. That's toward maybe the last quarter of this class. I'm just showing you here and here, and we did all that nonsense. And so remember, this is that 3-to-2 relationship of the pump. We're always ejecting out 3 sodiums and we're bringing in 2 potassiums. Think of it, that passageway is basically a 2-way passageway. It's got room for 3 sodiums and 2 potassiums. And you're going to see that. We're going to reverse that process when we get to something called the neuromuscular junction. Not a whole lot to say here. The cell adhesion molecules, by definition, are going to be on the exam you're taking. And so you do have to know that the desmosomes are the strongest, and they have keratin fibers, and they hold cells that have to be moving quite a bit. Tight junctions are about watertight. The more you have them, the less likely fluids can seep in between cells. We're not talking about straight through the cell, but in between the cells. The other cell adhesion molecules are gap junctions or connexons. They allow charges and signals to be carried. That's fine. That's really what you need to know. When they're talking about these reactions are a little bit different. This gives you a kind of idea of all the things they do. And we talked about some of these. A lot of those molecules are how fibers attach and tether the cells in connective tissue. Cells are always moving. Understand, your white blood cells are not sitting there waiting for an infection. Everything is moving, even within connective tissue. When you look at the slides of loose areolar connective tissue in the lab, or that were projected, it's like you got to get used to the fact that they're not just staying there doing nothing. They're constantly moving. The way they do is a process called rolling, and they're rolling over things and identifying them, or they're moving over them to get from point A to point B. If they weren't moving, again, the person would be dead. Okay, and they are, and a lot. Where do they move more? I mean, there isn't some magical signal. It's not like the cells call out to each other. It's about chemicals. When you stub your toe, twist your ankle, burn your finger in the kitchen, get a paper cut from opening an envelope, all of those things, when cells are damaged, they release chemicals. Those chemicals have a fancy name. They're called cytokines. And you'll learn about those. CYTO, all one word, KINES. Basically, they're signaling chemicals that spread very quickly, diffuse. And when the white blood cells receive that signal, they're attracted to it. Okay? And it pulls them toward that area. That's what's meant by cytokines. They're extraordinarily important. They're sort of the foundation of your immune system. So the white blood cells immediately, again, stub your toe, twist an ankle. The minute cells are disrupted, white blood cells will flock to the area. And that's what happens. Or if there's an invader like an infection, same deal. Okay. So all of those. And I mean, and again, they can do different things with regard to the junctions, intracellular signals, etc. Okay. Mostly contact signaling, and I sort of, it's like those short chains of carbohydrates. That's the identity feature. They touch and recognize in development and part of the immune system, that's self versus non-self. And there's a ton interactions between receptors and chemical messengers, ligands or chemical messengers. So we'll talk about receptors that are chemical receptors or ligand receptors. The terms are interchangeable, and they can do all sorts of different things. And we'll even learn why some things happen faster and some things are slower. Things happen faster when we move charged particles. Things happen relatively quickly. You're going to see a passage that I'm going to show you here that makes things happen fairly rapidly, okay, but not nearly as rapid as moving charged particles. And that is this particular kind of receptor. So here, different chemical responses, the receptor proteins change shape and activate, open and close things, excitability. It's better if I show it to you. It's called the G protein linked receptor. You do not have to know this just now. I want to give you an idea. This is how insulin works. This is how human growth hormone works. It's the way we talked about with signaling molecules or signal transduction. Something is so large it can't get inside the cell. It attaches to one of those receptor proteins and causes a shape change. And I have a really good animation that will show you all of these things down the way. And that shape change basically induces a cascade, a chain reaction within the cell. So that insulin or that human growth hormone, best examples, multiple changes inside the cell. In the case of insulin, it brings sugar in. That's just part of what it does. They're called G protein links. And the G stands for a molecule you already know about. Remember, we always talk about adenine and ATP. The G, the big G, GTP. They're very similar. Adenine and guanine, they both have those double carbon rings. They're called purines. They work similarly. We hear more about ATP, but on a cellular basis, I hear a lot about your G protein link. And they can do all of these things. So here's what they do. I mean, it's not a great, and this book has this. I haven't found one that's any better, but I don't like it. This whole chain reaction of things effectively. We'll do it this way. Here's the ligand. The ligand is the chemical. Interchangeable names. Insulin hits the receptor outside the cell, and basically they frame it like a relay race. It's like you're passing the baton and passing the baton and passing the baton. I was that kid who always dropped the baton. You didn't want me in the race. I am so slow. It goes on with my vertical leap of a half an inch. Someone who loves sports, I'm very unathletic. So the ligand touches the receptor changes shape, and effectively that shape change on the inside triggers a reaction in this protein, and it changes its shape, which goes along the interior and changes that shape, all of which require a certain amount of energy. And the TP, triphosphate, it's just another, those are high energy phosphates, just like ATP. So it's another potential high energy molecule. Eventually, it contacts what they call an effector protein or an enzyme. And what that does is it activates what's called the second messenger. The first messenger in the case here, ligand, is insulin. We're using that example. The second messenger, interestingly enough, is going to be one of two molecules typically. And you'll see both of them. One will be calcium ions, and the other, remember we talked about ATP, if the ATP loses two of those high energy phosphates, it's called AMP, adenosine monophosphate, and what it does is it changes its shape subtly. It becomes cyclic. In other words, it forms like a little, surrounds itself. So cyclic means forms a circle. So cyclic AMP, CYCLIC, we always just put a little C. That becomes what we call the second messenger. And the second messenger triggers a chain reaction. So if it's insulin, not only does it open the door for glucose to come in, it causes the cell to undergo mitosis, It directs other activities within the cell. Okay, it basically tells human growth hormone, take a break. I'm in charge. When I'm done, you can come in. And so it goes from the ligand, the first messenger, receptors by way of G proteins, activating an enzyme, and F second messenger. You're going to go over this multiple times. You'll see it in endocrine, particularly. Done. Thank goodness. Here you go. Something you should be familiar with. Good old parts of the cell. Yes, we're going to do that all again. Yeah, we'll do. And it's not all membrane-bound organelles. Understand we have both membrane and non-membrane-bound organelles. You probably had a lot of this stuff. Yeah, I already know what a ribosome does. I already know what a nucleus does. Fine. You're stuck learning it again. It's part of the curriculum. Get over it. If you did, you're going to hear it my way. And I'm doing it to understand the importance and the relevance of all of these. And basically, this is all about how your body works. So you have to understand the structure a little bit to understand the body. So, cytoplasm. It's basically what we call it sort of a solid gel. It's got stuff that is liquid, stuff that is gelatinous, and sometimes it's even solid. And so we end up with this sort of solid gel type of thing, going from pure liquid to semi-solid to solid things in it. The cytosol, more gel-like and watery at times, with things that are dissolved in it. And so just like gelatin, the more gelatin you put into the water, and eventually it'll firm up and you get jello, which is, I would say is a good example of semi-solid. Lots of cells have inclusions. We identify cells by what's in them sometimes. Glycogen, energy source. Pigments. In the nervous system, you see certain cells have pigments in them, and those pigments tell you where neurotransmitters are being made. We'll look at that in the second half of the course. Fatty droplets for storage. Different kinds of crystal structures that are in there. All of those play where you learned about one. Okay, melanin in the skin, right? So basically, the melanosomes, basically what do? They make melanin, they're little granules, they get absorbed by the keratinocytes, and you have pigmentation. It's not rocket science. It's just a chemical that gets in there and develops pigmentation. And so effectively, that's kind of like an inclusion. And yes, we have the machinery, so to speak, organelles. And they're either membranous or non-membranous. Okay. Even though we're going to look at the membranous first, yes, we're going to revisit shortly the mitochondria, because that's the one covered. We may not get to it, but the endoplasmic reticulum starts with the nuclear membrane, and there's this membrane passageway to protect everything that's being made that has to be encased in membrane. Anything in the cell that's going to be spit out, anything that's going to be incorporated in the cell's membrane, or anything that's going to be contained within membrane in the cell, like a lysosome, like a secretory vesicle. All of those transported through this thing called an endomembrane system. And the endoplasmic reticulum is really the next part after it leaves the nucleus. We're going to look at the packaging and shipping department, the Golgi apparatus. Send it down to the mailroom. It's the way it works. I try to use things that make it easy for me to remember. Whatever works for you is fine with me. Peroxisomes. We're going to learn about oxygen causes problems. Too much oxygen is not necessarily a good thing. Lysosomes, mentioned that already. Digestion and defense. You're going to see peroxisomes are more about defense against oxygen. It's what we call toxic derivatives of oxygen. And here, non-membranous, not membrane-bound. Ribosomes only become membrane-bound when a signaling molecule on messenger RNA that's telling it what protein, this is the protein you're going to make, it has a little signaling molecule that says, okay, you can stay here and make the protein. Oh, wait a minute. We've got to get you over and bind you to something called the endoplasmic reticulum because you're making something that's fragile. We're not going to just use it inside the cell. It's got to be contained in membrane. And that's the difference between what you'll see as a free ribosome and a bound ribosome. Okay. That's all the difference there is. The cytoskeleton, I alluded to that before. Sometimes there are things that should really, sometimes there are a physically fixed internal roadway where things are tethered to that. Yet a lot of those roadways allow things to move around. If you didn't know, mitochondria can move around. Okay. Again, there's a dynamic nature. Again, so you can have something that'll disappear here and be recreated over here. And these roadways are constantly appearing and disappearing. And we'll look at the cytoskeleton and the centrioles. They make, and you probably heard about this when you learned about mitosis and meiosis, that spindle. That's made of microtubules. That's another one of those things that can appear. Because if they didn't disappear, you wouldn't be able to tell if they, the old prophase, metaphase, anaphase, telophase. We're going to go over that again. Aren't you excited? My way. PMAT. Prophase, metaphase, anaphase, and telophase. Trying to help you. So here it is. The mitochondria, the power. I knew about this. They produce most of the cell's energy via aerobic oxygen requiring cellular respiration. You're going to see in a second it's all about the structure. Here, this is one of the early examples of that fan folding thing, those pleats I kept blabbing about. The fact that the membranes are fluid. You're creating surface area. You have two membranes. You have an outer membrane that's permeable, semi-permeable, and you have an inner membrane, but to create more surface area, in other words, the ability to produce unimaginably large amounts of ATP in a very, very small space, you end up with a membrane in here that we fan fold. Basically, all we do, and with my terrible illustration, is get the idea. What's happening in it? What's the importance of oxygen? So what happens? Bring it into the cell, the mitochondria, start processing sugar. Start breaking. But in order for it to generate the maximum amount of ATP, it has to have an entry. It has to get into the mitochondria. The entry is oxygen. So the glucose and the oxygen, effectively, remember glucose is T6. I'm not going to balance these T6, H12O6. And then effectively it's broken down, and all we have left to get rid of are effectively 6 and 6, like that. We end up with carbon dioxide, which water, which and a bunch of energy. That's really how we utilize it. That's the basic thing of what we call cellular respiration. Not so much the cells breathing, but it's doing an energy transformation, taking its basic energy source, glucose, in the presence of oxygen, generating a large amount of ATP, which is the way it does it, over the Krebs cycle, like that, and generating waste products that we can get rid of. And that's the great thing. We use those waste products very effectively in getting rid of them. The reverse of that is how we got that energy in the first place, called photosynthesis. We take, in this case, light energy, not ATP, in the presence of water and carbon dioxide in our environment, and with the help of plants, which have full chlorophyll, and what do we make? Carbohydrates, and it releases oxygen. Think of plant today. The oxygen you're breathing is coming from that plant. Thank you very much. You're doing a good job. Keep up the good work. It does. These neat green things. If you did not know it, most of the oxygen planet-wise comes from a microbe called algae. Remember, our planet's basically covered 80% in water. We just happen to be on the land part right now. So here they are, and other things maybe you didn't know. If you didn't know it, okay, it has its own DNA and RNA, and it's not the same as ours. There's something you'll learn about in micro called endosymbiotic theory. These are actually bacterial DNA, or looks just like it, and bacterial RNA looks just like it. Okay. Very, very different from ours. Yeah, and because of that, mitochondria can, and when we need them, make more. If you go to the gym and start a workout program, your muscles are going to get bigger. They're going to need more energy. You're going to be more mitochondria. If you have an injury, or all of a sudden you're tired of working out, or whatever, for some reason you can't get there for a while because you're busy with something else, all of a sudden those mitochondria will stop being made. and because they have a lifespan, they'll disappear. And the only reason they can do that is because they have their own DNA and RNA. They're like a little cell. They're not a living thing because they have to exist within that confine, but they were derived originally, we believe, scientifically, from an early form of bacteria. So you're going to see the DNA and the RNA and the ribosomes are a significant difference. We don't do it here. We do it in micro. Our cells do mitosis and meiosis. Bacteria do fission. Another thing that makes them look so similar. So here it is. You can see all that folding. The DNA is circular DNA, not good old double-stranded alpha helix. No. Got the A's, the T's, the G's, and the C's. Okay, but it's not. It does not have the alpha helix. It doesn't have that spiral staircase appearance. It has a lot of embedded proteins in it, and all this surface area is how we generate lots of energy. We'll leave it here, and we'll pick it up with ribosomes, as I left it with the other class, when you come in on Friday. Thank you very much. Bye.